Good morning, students, or good morning, or good evening, or good night. It depends on which which time you watch this lecture. Now I want to explain you the last immunology lecture, and I remind you that you should prepare both lecture number 8 and number 9 for the last immunology class. This lecture is a conclusion of all knowledge in immunology that you got during this semester. And the theme of our lecture is antibacterial, antiviral, and antifungal immunity, immunity in parasitic invasions, immunity in transplantation, cancer, immunity and age. In other words, we will talk how immunity changes in different conditions. In some infections caused by bacteria, viruses, or fungi, or parasitic organisms, how immune mechanisms change in transplantation of organs or tissues, what happens with immune response in cancer, in pregnancy, and what happens when when individual becomes elder. Let's start. The first one I want to remind that all pathogens that cause infections are classified into viruses, bacteria, parasites, and fungi. And all they cause infections or invasion, like parasites. And before we will start about features of antibacterial or antifungal immunity. Before it, we should discuss local immunity. Local immunity, it's immunity is protection of skin and mucosal membranes. Local immunity that includes skin and mucosal membrane is a mechanism or its barrier of innate immunity. You remember it from the lecture number 2. It's innate immunity. So it mediates it acts by three mechanisms. The first one is mechanical factors. The second one is flushing mechanisms of mucosal membrane, for example. And the third one are chemical factors. All these factors take part in protection of the patient, of the person. Mechanical factors of local immunity include skin, epidermis, keratin, mucosal membrane, lacrimal apparatus, and ciliary escalator. So all these mechanical factors provide mechanical protection of human organism against different pathogens. So if there is any wound, incisions on the skin or mucosal membrane, these are portals for entry of pathogenic microorganisms in human organisms. So it helps in causing of infection. Flushing mechanisms includes urine and vaginal secretions, because urine flush it flues bacteria from urogenital tract. Our vaginal secretions have the same action. Sneezing, coughing, swallowing reflex is also a flushing mechanism. You know that when we sneeze or cough, we discharge bacteria. It's adaptation, it's protective mechanism, protective process of our human organism when mucosal membrane try to discharge microorganisms. Movement of fluids across their surfaces like saliva also provides protection of local immunity, and washing action of tears. Tears also flew microorganisms from conjunctiva, and tears also have another action. Another action is also very important. Tears contain antimicrobial substances, for example, lysozyme, that is able to destroy peptidoglycan of gram-positive bacteria like Staphylococcus or Streptococcus. Tears wash away any microorganisms, debris on the ice surface. And the last one is the chemical factors. Chemical factors, it includes sebum and fatty acids in skin. Acidic environment is harmful for most bacteria. Gastric juice, it has low pH. That's why it also is harmful. For example, if the person has a normal stomach pH, such patient is not infected with Vibrio cholera. Cholera doesn't develop in the organism of such patients who has normal stomach pH. It develops just in those whose pH is acidic, is alkaline. Is not acidic but alkaline. You remember that Vibrio cholera like alkaline condition. Lysozyme, I've started to talk about it. It degrades bacterial cell wall. And antimicrobial peptides like beta-lysine with high quantity of lysine or arginine that disrupt plasma membrane of microorganisms. So all these factors are present in skin and mucosal membranes and provide local immunity or local protection of mucosal membranes of nasopharynx, vagina or urethra or intestine. All these factors provide local immunity of mucosal membrane and skin. Another question that we should discuss is features of antibacterial and antitoxic immunity. Factors of antibacterial immunity. So, which immune mechanisms are activated in bacterial infections? I hope you remember examples of bacterial infections. You had a colloquium this week, so I don't want to talk about it once more. I will talk about practical class. What are examples of bacterial immunity? Bacterial infections. So, which immune mechanisms are activated against bacteria? The first one is complement. It's known for you, and their functions are also known. It initiates chemotaxis and anaphylaxis by C3A and C5A components. It also take part in opsonization of bacteria. Opsonization is enhancement of phagocytosis. It's mediated by C3b component. It promotes killing of bacteria by membrane attack complex from C6 up to C9, and it provides activation of B cells. Another immune mechanism in bacterial infections is neutrophils. Neutrophils are the main cells that provide phagocytosis. You know that phagocytosis is mediated, is provided by neutrophils. These are the first responders in any bacterial infection. Dendritic cells. Dendritic cells are also main cells in antibacterial protection. They produce cytokines. For example, tumor necrosis factor alpha, interleukin 6, interleukin 1, interferon alpha. Some of these cytokines cause inflammation. And another function of dendritic cells is the presentation of antigen to CD4 cells, CD4 or T helpers that then activate CD8 cells. Macrophages that also provide phagocytosis. They also produce cytokines, pro-inflammatory cytokines and anti-inflammatory sometimes, like tumor necrosis factor alpha, interleukin 1, interleukin 6. They activate inflammatory response, inflammation, and they also present antigen to CD4 T cells. Macrophages and dendritic cells have the same function there. T cells mediate T cell immunity or cellular immunity. They respond to bacterial metabolites. Th1 CD4 cells that are T helpers. Responses are important for bacterial, especially intracellular infections, because they activate CD8 cells that are cytotoxic lymphocytes, CD8 cells, these ones. Cytotoxic lymphocytes release cytotoxic substances outside and destroy virally transformed cells or some tumor cells. Antibodies are also main mechanism of antibacterial protection. What are the main functions in antibacterial immunity? The first one is binding to surface structures of bacteria, fimbriae or capsule. The second one is blocking of attachment of bacteria to mucosal membranes to target cells. The third one is opsonization of bacteria for phagocytosis. Antibodies also take part in opsonization. Complement takes part in antibodies also take part there. Then promotion of complement action. You remember that complement is activated in the presence of immune complexes. Immune complex, it is a complex of antigen and antibody. And that's why antibodies take part in promotion of clearance of bacteria, clearance or killing of bacteria in a host organism, because they directly destroy the pathogen directly. And antibodies take part in neutralization of toxins and toxic enzymes. That's why we should say that antibodies is the only antitoxic immune mechanism that provides antitoxic immunity. Antitoxic immunity, what is it? Antitoxic immunity is produced in diseases whose pathogens release harmful exotoxins into the environment. For example, causative agents of diphtheria, botulism, tetanus, staphylococcus, and streptococcus. You remember these pathogens release very harmful toxin like tetanuspasmin or botulotoxin or diphtheria cytotoxin, exotoxin, or staphylococcus produce toxic shock syndrome toxin. And neutralization of exotoxins, of bacterial exotoxins, is caused by antitoxic antibodies. They are also called antitoxins. Antitoxic antibodies is the only immune mechanism that is active against bacterial toxins. So we also talk about antitoxic serum, for example, that is used against diphtheria, tetanus, botulism, gas gangrene. It's used for treatment of these infections. It's injected to block toxin in host organism. So it is about antibacterial and antitoxic immunity. Now let's talk about antiviral immunity. Antiviral immunity. We will talk about viruses in microbiology in lab lectures. Viruses are distinguished from bacteria, and these infections are distinguished from bacterial infections. That's why there is some differences. There are some differences in antiviral and antibacterial immunity. The first main mechanism of antiviral protection is interferon. Interferon. I hope you remember it from the second class. We talked about it. Interferon causes inhibition of enveloped viruses. It initiates the antiviral state in surrounding cells. It blocks local viral replication and activates systemic antiviral responses. So interferon, there are three types of interferon: alpha, beta, and gamma. The most effective interferon against viruses is alpha interferon or leukocyte interferon. So it also used for treatment of viral infections, for treatment and prophylaxis. The another immune mechanism that is active in antiviral protection are natural killer cells or NK cells. They activate macrophages. And another their main mechanism, they kill virus-infected cells, especially enveloped viruses that are present sometimes present intracellularly inside the cells. The third immune mechanism of antiviral protection are macrophages and dendritic cells. It's also present in antibacterial immunity. Macrophages filter viral particles from blood. They also inactivate opsonized virus particles. Immature dendritic cells produce interferon alpha. We have just said that its main antiviral immune mechanism, immune protein. Dendritic cells and macrophages present antigen to CD4 T cells. So presentation of antigen is also main function of macrophages and dendritic cells. The another immune mechanism of antiviral immunity is T cells. T cells recognize viral peptides on cell surfaces. CD8 cytotoxic T cells, I remind you that these are T killers. Don't be confused with natural killer cells. Natural killer cells are also called null cells. Null cells, they don't have any CD markers on their surface, but T cells, these are lymphocytes. CD8 cells that are T killers respond to viral peptides, so they are activated in the presence of viral peptides. CD4 Th2, these are T helpers. Responses are important for the maturation of the antibody response. You remember that Th2 activate antibodies, activate B cells that produce plasma cells for production, for synthesizing of antibodies. So we can say that Th2 cells, is responsible for activation of antibody production. And sure, antibodies also take part in antiviral protection. At first, antibodies neutralizes extracellular viruses. It is very difficult, almost impossible, to neutralize intracellular viruses, the viruses that are present intracellularly in host cells. This process of killing of intracellular viruses is mediated by natural killer cells, mostly natural killer cells, and sometimes by CD8 cells. Then antibodies also opsonizes virus for phagocytosis. They provide successful phagocytosis. Then antibodies promotes killing of target cells by the complement and antibody-dependent cellular cytotoxicity. Promotes killing of infected human cell by activation of complement, because you know that complement is activated in the presence of antibodies, IgG, and antibody-dependent cellular cytotoxicity. What else? Antibody also block viremic spread to target tissues. They cover the surface of viruses and block it. IgG is a more effective antiviral immunoglobulin than IgM. And another one, that is secretory IgA, is important for protection of mucosal surfaces. You know that secretory IgA is produced on the surface of mucosal membrane, so it provides local protection. The another one is antifungal fungal immunity. Antifungal immunity. What happens with immune system, which immune mechanisms are activated in fungal infections. You remember that fungi cause infections, cause diseases that are called mycosis. You remember there are some groups of mycosis. What are they? These are superficial mycosis that includes keratomycosis and dermatomycosis. Then subcutaneous mycosis that infect subcutaneous tissues and muscles, and deep mycosis that infect internal organs, lungs, for example, liver, and spread within the human organism. So immunity in fungal infections is mediated by some immune mechanism and depends on the type of infection. If we are talking about superficial mycosis, dermatomycosis, and keratomycosis, an immune response doesn't develop at all. If you remember, superficial mycosis infect outer layer of skin, hair, and nail. They may invade. Dermatomycosis may invade deeper, but they don't contact with immune cells, so immune system doesn't respond. Immune cells don't contact with fungi. But in deep mycosis and subcutaneous mycosis, immune system responds to these infections. What are main factors of antifungal immunity in deep mycosis and subcutaneous mycosis? The principal mediator of innate immunity against fungi is the neutrophils. Neutrophils liberate fungicidal substances such as reactive oxygen species and lysozyme enzymes that destroy fungal cells. Neutrophils also phagocytose or engulf fungi for intracellular killing. Fungi that are present intracellularly in macrophages are eliminated by the same cellular mechanisms that are effective against intracellular bacteria. What are these cellular mechanisms? These are natural killer cells and cytotoxic CD8 cells. So main immune mechanisms in antifungal immunity are neutrophils that produce fungicidal substances, lytic enzymes, and provide phagocytosis, and then natural killer cells and CD8 cytotoxic cells. The next type of immunity that we should talk about, it's immunity in protozoa and parasitic invasions. Protozoa and helminths parasites that enter the bloodstream or tissues are often able to survive and replicate because they are resistant to host innate immune responses. So protozoa, we don't study protozoa in microbiology, you had to study it in biology, like malaria and other amoeba, dysentery. Helminths or parasites can be survived and replicate. They are very resistant to immune response of human organism. They are less immunogenic. Protozoa develop cysts that are resistant to immune response. That's why it is low. Immune response is low. Antigen masking is an effective form of immune response evasion by some parasites. Parasites in human hosts are usually resistant to complement. Macrophages can engulf protozoa, but the tegument of helminthic parasites, tegument its outer layer, tegument of helmintic parasites makes them resistant to the lytic effects of both neutrophils and macrophages. So we can see that such immune mechanisms as complement, neutrophils, macrophage, and phagocytosis are not effective against protozoa and parasitic helminths. Protozoa that replicate inside cells and lyse host cells stimulate specific cytotoxic lymphocytes responses. So the first immune mechanism that is effective against protozoa organism and parasitic invasions are cytotoxic lymphocytes, CD8 cells that release cytotoxic substances outside to kill these cells. And another main mechanism of anti-protozoa and anti-parasitic immunity is immunoglobulin E, IgE. IgE antibodies and eosinophils mediate defense against many helmintic infections. So, cytotoxic lymphocytes, IgE, and eosinophils are effective against protozoa and parasitic invasions. The next one that we should discuss, that we should talk, it's immunity in transplantation. Firstly, I hope you know what is transplantation. Transplantation is process of transfer of organ or tissue or cells from one organism to another organism, from donor to recipient. Actually, transplantation is a very difficult process. It's a very difficult process. Why? Because you remember that any individual has his or her antigens. What are antigens? You remember that humans have ABO antigens, rhesus antigens, CD antigens that is present on lymphocytes, and tumor antigens. And another one that is very important, the most important for successful transplantation, it is human leukocyte antigen, HLA. And major, that is example of major histocompatibility complex, MHC. There are two types of MHC, class 1, class 2. Please look for in lecture number 3 what is MHC. I have explained it. MHC is personal antigen, specific antigen for every person. That's why transplantation is a very difficult process because of activation of immune response, activation of immune system, immune mechanisms against transplanted organ or tissue. So what happens? What happens in transplantation? Antibodies in recipient organism bind to donor endothelial cells in transplanted tissue. For example, let's imagine that we transplant kidney, and you understand that in every organ, in every tissue, there are a lot of blood vessels that are composed of endothelial cells. So antibodies of recipient bind to endothelial cells of transplanted tissue or organ, and they form antigen-antibody complex. It activates the complement system, causing inflammation. You remember C3A, C5A causing inflammation, and massive thrombosis because of inflammation in the capillaries, which prevents the vascularization process of formation of new blood vessels of the graft. So let's look at this diagram and this picture. It is we can see blood vessel with endothelial cells. When we transplant organ or tissue in recipient, antibodies of the donor bind to endothelial cells because there are antigens, blood group antigens here. So they activate, there is immune complex, they activate complement. C3AC5A cause inflammation, endothelial damage because of due to inflammation, and thrombosis. And new blood vessels are not formed. Graft, it is a transplanted organ or tissue. Graft is rejected within from minutes up to hours because vascularization is rapidly destroyed. Oxygen doesn't reach tissues and graft is rejected. It occurs because the recipient has pre-existing antibodies in circulation against the graft. Firstly, we should say that these antibodies must be present. It happens in the secondary and every following transplantation. And this process, if you remember, it's example of cytotoxic hypersensitivity. It's from lecture number 4. When it develops in blood transfusions, for example. So pre-existing antibodies could be induced by prior blood transfusion, multiple pregnancies, or prior transplantation if transplantation was made before it once or sometimes. What about organs that is more susceptible for graft rejection? This is kidney. Kidney is the most susceptible to hyperacute rejection. So it's about transplantation. That's why it is very important. It's very important to control antigens of donor and recipient at first, and then there are special immunosuppressive drugs that are used in transplantation for inhibition of immune response. I've said about immunosuppressive drugs in lecture number 8 in previous one. The another one that we will talk about is immunity in tumors. Firstly, what is tumor and what are tumor cells? Tumors arise from accumulated genetic mutations. Tumor may occur in any human cell due to mutation, modification, and it causes modification of human cell. Human cell receive or gets new antigens due to modification, so it becomes antigen for human immune system. Tumor antigen is called T antigen. It may be classified into tumor-specific antigens that are present only on tumor cells and not on any normal cells, and tumor-associated antigens that are present on both tumor cells and also on some normal cells. What are main immune mechanisms in anti-tumor immunity? These are cytotoxic lymphocytes, CD8 cells. They are major immune defense mechanism against tumors because they release cytotoxic substances and kill transformed human cells. Transformed or virally transformed natural killer cells is another one. They are capable of destroying tumor cells without prior sensitization. It is a first-line defense against tumor cells. Natural killer cells are activated earlier, faster than cytotoxic lymphocytes. And their action is almost the same. Natural killer cells release lytic substances, cytolytic substances. Activated macrophages is also a main anti-tumor immune mechanism. Activated macrophages may kill tumors by mechanisms similar to those used to kill microbes. For example, by production of reactive oxygen metabolites or by secretion of tumor necrosis factor. So tumor necrosis factor is also main anti-tumor mechanism, anti-tumor protective mechanism. Interferon gamma. Interferon gamma, you remember that interferon are cytokines that are produced by activated immune cells. There are three types of interferon: alpha, beta, and gamma. We have already said about interferon alpha in antiviral immunity. In anti-tumor immunity, we should talk about interferon gamma, that is secreted by T cells and natural killer cells, and it's a potent activator of macrophages in anti-tumor immunity. I have said that there are two types of tumor antigens. These are tumor-specific antigens and tumor-associated antigens. They are very important. They are useful as tumor markers released only from tumor tissues, and they are specific for a given tumor type or organ. They present in all patients with tumor, and that's why can be used in diagnosis of tumor. Their detection of tumor antigens is used in detection of cancer. They are also called oncological markers, oncomarkers. Detection of oncomarkers is used for diagnosis of tumor. It's recommended for for risk group patients who had a tumor or whose relatives have tumor, and for adult persons, because adult persons are susceptible for tumor development for cancer. The another one that we will discuss, it's immunity in pregnancy. Immunity in pregnancy. It's very interesting. In organism of pregnancy woman, we can see wonderful changes. Firstly, the immune system significantly changes during pregnancy. What happens? Remember, please, that normal implantation and parturition, it is early pregnancy, normal implantation, early pregnancy, and parturition or birth of the child, of the baby in delivery, needs activation of pro-inflammatory environment or pro-inflammatory mechanisms. So in early pregnancy and during the delivery, during the birth of the child, in the first trimester, in the end of the pregnancy, pro-inflammatory substances, pro-inflammatory cells are activated. While normal placentation and fetal growth need a tolerogenic environment. Tolerogenic means equal amount of pro-inflammatory and anti-inflammatory. So normal fetal growth, normal placentation needs equal amount of pro-inflammatory and anti-inflammatory. The first trimester of the pregnancy is dominated by natural killer cells and alternatively activated macrophages. Complement is activated in large amount. Neutrophils, I will show you then as a scheme what is activated in different trimesters. Pro-inflammatory factors, for example, C-reactive proteins present in the plasma of pregnant women. Actually, C-reactive protein is produced in some infections, in inflammation, and in different injury of human organism. But you should remember that C-reactive protein is produced, its high level is observed in pregnancy woman. It doesn't mean that pregnancy woman has infection. No, it's normally for pregnancy woman. Then I have said that normal placentation, it's the middle of the pregnancy needs tolerogenic environment. It means that amount of pro-inflammatory and anti-inflammatory substances must be the same. So we can observe decreased Th1 to Th2 balance when anti-inflammatory action of immune cells is observed. It is used to protect fetus against pro-inflammatory mechanisms of mother's immune system. In peripheral blood, regulatory T cells are increased during early pregnancy. T cells increased in early pregnancy because they provide inflammation also. They have required for the maternal immune system to tolerate a fetal allograft. Let's look at the scheme. Early pregnancy, during pregnancy, or fetal growth, and delivery phase. Early pregnancy, it's implantation, you can see. During pregnancy, development of the fetus, and delivery phase, it's the birth of the baby, of the fetus. And look here. In early pregnancy, pro-inflammatory environment or pro-inflammatory substances, all are they here. So amount, you can see that its amount increases in early pregnancy. What's amount? Neutrophils, macrophages, dendritic cells, and interleukins like pro-inflammatory interleukin 6, 1, interferon, tumor necrosis factor. Natural killer cells, immature cells. Then, during the pregnancy, immune system mother doesn't need pro-inflammatory environment. You can see that its amount and activation of inflammatory cells and substances decreases. So here we can see anti-inflammatory predominant of anti-inflammatory substances. These are anti-inflammatory interleukin like 10, for example, transforming growth factor beta. These are T regulatory cells. And then for successful delivery, immune system of mother once more needs increasing of pro-inflammatory substances. You should remember about dynamics of changes in immune system of pregnancy woman. We have just said about changes in immune system of pregnancy woman. Now let's talk about features of immunity in aging. How immune response changes during the age, during the life. The efficiency of the immune system declines with age, but this is variable among persons. Sure, level of immune system is distinguished in every person. Some people are infected very often, very frequently by different viruses or bacteria and other microorganisms, but I know those people who are susceptible, who are infected, who are sick, very rare. But there is the same dynamic, the same mechanism um, in all people. Immune response decreases with age. We know that elder person are more susceptible to infections and cancer and autoimmune disease, more susceptible than young patients. What happens? Non-specific defense during age becomes less effective due to decreasing of lysozyme, C3 complement and beta-lysine, and decreasing of macrophages, poor chemotaxis of these macrophages, and poor engulfment and digestion of antigens. Then ability of the body to make antibodies also decreases, so amount of antibodies also decreases. We also can see poor antiviral and anti-tumor defense in elder persons, in elder patients. T cells proliferate much less, decreasing of amount of natural killer cells, and their activity is observed. Autoimmune disorders are increased in elder adults, in elder persons. And you remember that thymus gland atrophies throughout life. After puberty period, it atrophies. What is result? What is result of these processes in the immune system? Common infections are often more severe with slower recovery and decreased chances of developing adequate immunity. Elder patients are more sensitive or more susceptible to infections. These infections are more severe than in a young person. Autoimmune diseases occur and sure cancer, because anti-tumor, anti-cancer immunity is not effective, because T cells are not so proliferated, not so active. Other cells, macrophages, are not so active. These cells produce main anti-tumor substances and activate anti-tumor cells. So that's all. It was the last immunology lecture. Thanks for your attention during this semester. Thanks for your attention. Take care of you, and don't forget that you should prepare both lectures number 8 and number 9. So you can see that lecture number 9 is less than lecture number 8. Goodbye. Thanks for your